Welcome to this lesson on the Krebs cycle, also known as the citric acid cycle. This is a crucial process in cellular respiration that takes place inside the mitochondria of our cells. The Krebs cycle is responsible for extracting energy from the food we eat, specifically from a molecule called pyruvate. Throughout this video, we'll explore how this elegant cycle works to power our cells and keep us alive. So what exactly is the Krebs cycle? It's a series of chemical reactions that work together to extract energy from pyruvate molecules. This cycle occurs in the mitochondrial matrix, which is the innermost compartment of the mitochondria. You might also hear it called the Citric Acid Cycle or TCA Cycle, which stands for Tricarboxylic Acid Cycle. All three names refer to the same important process. Let's understand where the Krebs cycle fits in the bigger picture. It all starts with glycolysis, which breaks down glucose and produces two pyruvate molecules. These pyruvate molecules then go through a transition reaction where they're converted into two Acetyl-CoA molecules. Finally, these Acetyl-CoA molecules enter the Krebs cycle, where the real energy extraction begins. Think of it as a production line where each step prepares the molecule for the next stage. Before we dive into the steps, let's meet the key players. First is Acetyl-CoA, a 2-carbon molecule that enters the cycle. Next is Citrate, a 6-carbon molecule that forms when Acetyl-CoA joins the cycle. Then we have Oxaloacetate, a 4-carbon molecule that regenerates at the end to keep the cycle going. Finally, carbon dioxide is released as a waste product that we eventually exhale. These molecules work together in a perfectly coordinated dance. Now let's walk through the first 3 steps of the cycle. Step 1 is the formation of citrate. Here, Acetyl-CoA combines with oxaloacetate to form citrate, a 6-carbon molecule. Step 2 is isomerization, where citrate is rearranged into isocitrate, still maintaining 6 carbons. Step 3 is the first oxidation reaction. Isocitrate loses 1 carbon as carbon dioxide and produces NADH, leaving us with alpha-ketoglutarate, a 5-carbon molecule. This is where energy extraction really begins. Continuing with steps 4 through 6. Step 4 is the second oxidation. Alpha-ketoglutarate loses another carbon as carbon dioxide and produces another NADH molecule, forming succinyl-CoA with 4 carbons. Step 5 is called substrate-level phosphorylation. Succinyl-CoA is converted to succinate and produces GTP, which is equivalent to a TP. Step 6 is the third oxidation, where succinate becomes fumarate and produces FADH2. Notice how each step extracts a little more energy. Let's complete the cycle with the final steps. Step 7 is hydration, where water is added to fumarate to form malate, still a 4-carbon molecule. Step 8 is the fourth and final oxidation. Malate is oxidized to regenerate oxaloacetate, producing 1 more NADH molecule. And here's the beautiful part. Oxaloacetate is now ready to accept another Acetyl-CoA molecule, starting the cycle all over again. This is why it's called a cycle. It continuously regenerates itself. Let's look at what 1 turn of the cycle produces from a single Acetyl-CoA molecule. The cycle generates 3 NADH molecules, which are high-energy electron carriers. It also produces 1 FADH2 molecule, another electron carrier. Additionally, 1 GTP molecule is made, which can be converted to a TP for immediate energy use. These products might not seem like much, but they're crucial for the next stage of cellular respiration. Remember that each glucose molecule produces 2 Acetyl-CoA molecules, so the cycle runs twice per glucose. This means we need to double all the outputs. From 1 glucose molecule going through the Krebs cycle twice, we get 6 NADH molecules, 2 FADH2 molecules, and 2 GTP molecules. These electron carriers will carry their high-energy electrons to the electron transport chain, where the majority of a TP will be produced. Let's recap the most important points. The Krebs cycle completes the oxidation of glucose that started with glycolysis. The NADH and FADH2 molecules produced here carry electrons to the electron transport chain, which is the final stage of cellular respiration. This entire process takes place in the mitochondrial matrix. And don't forget, carbon dioxide is released as a waste product, which we breathe out. Master the Krebs cycle, and you've mastered a fundamental part of how cells produce energy. Thanks for watching.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.